So, very good afternoon. Right? Audible? Right. Okay. So, very good afternoon, everybody. Today we discuss the unit third, that is the means black third structure of pollen grain and ovule. Simultaneously, we discuss the seed development after fertilization, how the seed will be developed, and heterosis also in this log 3. Okay. That last class we discussed that one, the seed treatment and the remaining marketing and labeling that we are to discuss last class. Okay. So now here the black card, here 3 units are there. 3 units. That is the structure of pollen grain and structure of ovule. That is unit 7 and unit 8. This will have hybrid seed production. And that is the 9th one is the seed development and heterosis. We have to discuss. In the first unit means 7th unit, we discuss the structure of pollen grain and ovules. Different types of the ovules are there, pollen viability and structure of different pollen grains, pollen storage also. We have to discuss here one by one. Means here, that is the what is pollen grain? So structure of pollen grain here. What is the pollen grains? We know that one, which is the reproductive process. This one is the male part. Pollen grains are microscopic structure that is vary in size and shape, different depending on the species. Okay. So this one is produced from the anthers, which is developed inside the anthers. In some they are tiny, while other are the egg-shaped structures are there. Although too small to see individually, they can be easily seen by the naked eyes. That is the based on their size and more, means large in size. A pollen grain hardly looks extremely durable body, and it has a taut outer coating. This hard coat effect great protection from the harsh outdoor environment. Means these outer exine means outer the protecting layer we call as the exine. This exine is very hard in structure and which is protect from the physical and chemical forces from the environment. This is the protecting layer. This is the important because inside it is top shell layers, 2 cells are there. That is the tube cells, which is the eventually became the pollen tube, and generative cell, which is contain the that is the nucleus, means sphere nucleus are present, that we call as the male nucleus for the fertilization. Well, there are the 3 main components of the pollen grains are there. What are the inside of the pollen grain is made up of the cytoplasm. This one is the germplasm or cytoplasm there. This fluid media houses the unformed living cells, keeping them moist and alive. The outer shell is made up of 2 layers. The inside layer is aptly named as the intile. And this one is it is made up of the cellulose. So a common component of the cell wall of the plant cells. Here I shown to the pollen grain. See here, I shown to a picture. Structure of pollen grain here. So I share to one picture. See. So this is the structure of pollen. Clear? That is the outermost layer is the that is the exine, and inner one is the made up of cellulose intine, and that is the cytoplasm is there. Inside the cytoplasm, the tube cell and connective cell, generative cell is nothing. Okay, the tube cell there goes and the generative cell to act as a the male sperm. Okay. The outer layer is known as the exine. We know that one. This is a highly sophisticated and complex outer layer is stitching compound known as the sporopollen. Sporopollen is very hard structure of chemical compound, waterproof resistance to the deterioration and very stiff. This usually is basically one of the nature's most advanced polymer. It ensures that the tender cell inside have a strong chance of survive. So that's why the pollen grain is very hard in structure and it will sustain long durable time. And the often time of exile has folds, creases, and spikes rising from its surface. So see here that picture, that is the photo. I shared that photo. They have some spines like structures are there, appendages are there, spikes are there, prolines. That is the ornamental is present. These features add to the protective nature of this layer. They also play an important role in the mobility of the pollen grain, making it is more likely that they will stick to the legs of insects as catch the wind for the pollination. Okay? That is the structure of the pollen grain. Clear? Now, so coming to the structure and types of megasporangium, means female gametophyte. So the process of formation of megaspore from megaspore mother cell by the meiotic division, that process we know as the megasporogenesis, which takes place in the ovule. Means the megaspores formation from megaspore mother cells. Four megaspores are developed from the each megaspore mother cells during the meiotic division. That development process we call as a megasporogenesis. The ovule is considered to be an integumented megasporangium. The ovule consists of the stalk and the body. The stalk is called funicle. One end of the funicle is attached to the placenta and other end by the ovule body. So this one also I see here. So this is the female gametophyte, right? Here, female gametophyte. There is a picture. There is the integments are there. So sometimes the funicle get fused with the body of the ovule one side and forms a rigid known as a rocket. The body of the ovule shows two ends, the basal end, often called as the chalazal end. The upper end is called as a micropylar. So here basal part and upper part is there. Basal one we call as a chalazal and upper end is called as a micropylar. The main body of the ovule is covered with one or two envelopes, which is called as the integments. See here, integments are there. The opening place is the micropylar upper region and the lower base, that is the chalazal region. So these leave an opening at the top ovule called as a micropyle. The integments close a large parenchymatous tissue known as the nape. The residual part of the nucleus in the mature seed is called as a perisphere. In the center of the nucellus is situated a female gametophyte known as embryo sac. So here some of the integments are there. The ovule in relation to integments, that is the uni-integument and the bi-integument area caruncle attachment. So here uni-integuments, the ovule with a single integument, that is the Gamma-fetalis dicotyledons. All are the uni-integments, means a single integument is present. By integumentic ovule with the single means two integments are present. Aerial, that is the another structure of the ovule is covered, means seed covered, another type, that is the a collar-like outer growth from the base of the ovule and form the third integument. So that is the aerial is found in the leech and carancule. This one is the most important. Carancule is also formed as a outer growth of the third outer integument in the micropylar region. Carancule is commonly in the ovules of Euphorbiaceae, example castor. Castor seeds, they have the white structure of the in the micropylar region. Seeds, they have the crown-like structure or collar-like structure on the upper region at the end of the top. That is the carancule. And another one, ategmic. Ategmic means in some parasitic like Loranthus, discum, there is no integument, such as called as a integument or ategment types of ovules. Here, the different types of ovules are there depending on the shape and the orientation of the ovule of angiosperms. Shape and orientation means they are the position of chalazal and micropylar ends. These depending on these positions, the types of ovules here, different types are there in the angiosperms. First one is the arthrotrophous ovule. So arthrotrophous ovule means I shown the diagram here. That is the arthrotrophous ovule only. So What is the arthrotrophus means? The microphylla and the chalaja ends and funiculars, they are the straight line, which is possess the straight line. This is the most primitive type of the ovule in the angiosperms. This one is the most primitive of the angiosperms. Means chalagal, microphylla, and funicle, which is here the straight line. That one we call as the arthrotrophus ovule. And another one is the anatrophus ovule. Means the ovule of the body is completely inverted into 180 degrees, so that the microphylla and hilum come to be very close each other. That one is the anatrophus ovule. Means completely half portion which is curved, inverted. That is the anatrophus. So hemianatrophus ovule means the ovule turn at 90 degrees angle upon the funicular body of the right angle to the foot. Right angle to the funicule. That can be called as hemianatrophus. And comphylotrophus. Comphylotrophus means the ovule is circle more or less at a right angle to the funicule. So microphylla end is slightly bent downside. Slightly bent to the downside. That is the comphylotrophus. And amphitrophus. So and in amphitrophus, that is the ovule totally curvature is occurred. Ovule, which is curvature mode, and the embryo sac becomes curved like horseshoe shape. That is the lemma and poppy alice. This is the amphitrophus ovule. And another one is the sarcinotrophus ovule. In the sarcinotrophus ovule, the ovule is initially arthrotrophus, but became the anatrophus due to the unilateral growth of funicle. The growth continues till the ovule one again becomes arthrotrophus. As a result, funicle completely surrounded the body of the ovule. Funicle looks like another layer of the tegments, so that is the which became to come as a arthrotrophus. Okay. Example, opentia. This is the and formation of megaspores means megasporogenesis. Here, the ovule or the megasporangium develops as a small protuberance of the placental tissues. It is very young ovule in single hypodermal cell is differentiated as archisporial cell. The archisporial cell may directly function as megaspore mother cell. The many are either divide pericanally to form an outer parent partial cell and inner sporogenous tissue. The sporogenous cells directly behaves an megaspore mother cell. The diploid megaspore mother cell enlarges in size and divides by meiosis to form a linear tetrad are 4 applied megaspores. The occasionally T-shaped or inverted T-shaped tetrars are formed that the megaspore first cell of the megasporophyte. This is the megasporogenesis. And the next one is the pollen storage. Pollen storage means what is pollen here? The pollen is very useful source of food, diverse alleles within the gene pool, and so may be effective propagule for gene bank. The case of the pollen storage and shipment and potential for its the immediate use provide researchers with the increased option when they designing their breeding program. Method for pollen collection, desiccation, viability test, and languidity assessment have been developed for many species of interest and have related to and revealed to critical importance for increased languidity by using high quality pollen desiccating sufficient in the rapid manner. So the viability assessments are development upon adequate rehydration and use of viable strains. In vitro germination, assess in vivo pollination experiments, pollen preservation in gene banks will likely be implemented as the standard procedure for hardening and assessing it as development. Advantages to the use of pollen. So, what is the advantages here? The gene bank pollen can be made available to breeds upon the request for a tree species. This alleviates the need for growing the male parent in the breeding orchids. It allows for a wide hybridization across seasonal and geographical limitation and reduce the coordination required synchronize the flower and pollen availability for use in the process. With adequate pollen viable, one can also load additional pollen into stigmas to increase pollination and yield. Pollen is available for research progress. As a single cell, pollen provide as a simple model system for research on the conservation. The storage of pollen with the gene bank also ensure it is availability year around for the basic biology and allergy research programs. Okay, and so another important here, the pollen capture diversity within the small sample sizes and documentation is available for long term, which is survive the pollen from many diverse species. Here we have to discuss that one. The pollen also serves as the source of genetic diversity in the collection, where it is hard to maintain diversity with the species. So another advantage, the pollen can be shipped internationally, often without threat of disease transfer. So this one. So I shown to table for the preservation means pollen gene bank stored in the liquid nitrogen. And process are there. Okay. So see here. This is the first one is the different species and storage time, and the temperature means liquid nitrogen pollination viability test, and another one is the reference here to discuss in the on the table. And another one is the disadvantages. What are the disadvantages in the pollen storage? First one is the limited pollen production in some species. Very little amount the pollen grain produced in some species. The primary limitation in their routine implementation of the pollen storage within gene bank is the difficulty in the obtaining adequate quantities of pollen for many species. So, labor intensive collection and processing. For some species, pollen is readily available, but the resource to accumulate and process enough pollen for routine storage and distribution are inadequate. And another disadvantage that is the standardized, no standardized processing or viability and test protocols. Either processing and viability testing method have not been documented and standardized in particular manner, so that the seed testing and regeneration of aged pollen. So seed regeneration can often be performed directly using the seed sample in storage process. For the pollen associated mother plants are necessary to the replenish pollen supplied when the quantities are depleted have deteriorated. These are the some disadvantages in the pollen preservation or pollen ban. And another one, pollen viability. See here, I am shown to another chair. So see here, pollen viability test. Here, the pollen tube will be generated. What is the pollen viability test here? Yeah. So in the staining process, one commonly used vital stain is the fluorescent ester, and fluorescent diestate. This test measure the membrane integrity. So pollen grains fluorescence green when a cellular ester is the FDA. FDA means fluorescence diestate. This assays depend on upon the functional membranes, the osmoticum of the FDA means the fluorescent diestate. Staining solution is the critical. Stain is often dissolved 10% or to 20% super solution containing boric acid and calcium nitrate to minimize plasmolysis and membrane leakage. Another one is the comparison of comparisons of viability of determined through the use of FDA or tetrazolium and those obtained using in vitro germination or in vivo tetrazolium tests reveal the consistently high correlation provided pollen is adequately rehydrated prior to testing. FDA has occasionally been shown to give the false negative result where the viable pollen appear dead. This is the pollen viability. So the several tetrazolium based staining are available for the testing pollen viability. The test was shown to give the most dependable results in the comparisons trial using the plume pollen. The tetrazolium test measures the ability to reduce the colorless tetrazolium to colored formans. So, which is to identify the pollen that has capacity for the oxidative metabolism by the Heiser and Marison in 1964. So many other vital staining have been developed and proposed over the past 50 years, such as the Alexander Estoa-Karamin, aniline blue, and XGEL, which is successfully identify the viability of pollen grains. Okay, so this is the structure of pollen grain and structure of megasporogenesis means formation of megasporosynthesis and structure of megaspores. Types of megaspores means ovules. Different types of ovules we have to discuss and seed banks and another one is the pollen viability test. Right? Now coming to the principles of hybrid seed product. So what is means hybridization here? The hybrid is produced by crossing between two genetically dissimilar pairs. That one is process called as the breeding, hybridization. The pollen from male parent will be pollinated and fertilized, set to seed in the female to produce F1 hybrid seeds. For producing the hybrid crossing between two parent in this is important. The crossing process will be result heterosis. In nature, to create genetic viability and for its wider adoptions, different environmental conditions. So flowering plants has adopted many mechanisms for cross pollination. So cross pollination results in genetic heterogeneity and show the wider adaptations. Flowering plants have evolved a number of devices to encourage the cross pollination. Some steps are there which are involved in the hybrid seed production, hybridization steps. So first one, the selection of parents. These steps are more important to produce the hybrid seeds. Selection of parents is the first one. This one is the very important step for the producing the hybrid seeds. So means the selection of parents should be done with the great care and taking into consideration of objectives so that the chance to select the desirable genotypes would be more from the segregating population. Desirable characters we are to select. So the possible parent types must be selected from the local types because they are best to adaptable. So while selecting the parents or the important characters to be combined must be kept uh, in the view. And selfing the parents, self. In this second step, it is essential for the elimination of the undesirable character and also obtaining the homozygous individuals. So that's why the selfing to be needed for the self-pollinated crops, while the cross-pollinated crops will be performed artificially. So self-pollinated crops called cross-pollinated crops are there. You know that one here self-pollinated is not necessary for the selfing. So here cross-pollinated crops inflorescence should be included in the bags before the anthesis to avoid the cross-pollination. So that's why here inflorescence, total inflorescence should be covered a polythene bag or cover before the anthesis. In the crops like castor, all the inflorescence closed in the bag. And in maize, selfing is done by collecting the pollen from the tassel and dusting on the silks of some plants. Another step is the emasculation. So this is very important step in hybridization process. Emasculation is the removal of male or the bisexual flowers before the anther rip. Shed the pollen on the stigma to prevent the self-pollination. So another one is the forced open methods. In this method, the flowers are opened by the force before it opens naturally, and the anther are removed with the forceps. This technique is useful in the large size of flowers like tomato, brinjal, and chilies. And another one is the clipping method. So in the clipping method, this technique is usually used in cereals like rice, wheat. So in this method, that is the 1/3 portion of the stickles that is are cut with the scissors and anthers are removed with the force. And the ring cut method also we have to keep. and keel rupturing method and hot water treatment also. These methods are for the emasculation process are the hot water treatment. Here, they force it in time to take and tedious the plants having the small size cover such as rice, jawar, and remaining plants. In such cases, dipping the panicle in the hot water for a defined period of time, which is the desired temperature, is effective. This makes the pollens inviable without affecting the stigma and ovary also. The panicle cut can be dipped the thermos flask containing hot water. The treatment of water is time depending upon the different crops, which is around 45 to 50 degrees for 10 to 2 to 10 minutes. And cold water treatment, chilling method, cold water treatment also around 4 degrees to 6 degrees temperature or cold water and the particular time. And the relative humidity method, hot air method, and chemical alcohol treatment. These all are there for the stop the cross pollination. These methods are there, including the emasculation. So male sterility method. In crops like maize, jawar, bajra, and sunflower, the male sterility occurs due to the cytoplasmic and genetic causes. The emasculation is not need to do in such cases. So another important aspect, self-incompatibility. So crops like tobacco, cabbage, and cauliflower, pollination with the desired pollen is possible without doing emasculation due to the presence of self-incompatibility. This is the very advanced method and incompatibility. So bagging, another step is bagging. The selection male and female flowers have been bagged separately to prevent the contamination is staminated flowers and cross pollination in the pistil flowers. Bagging has to be done in variability and precious evening of the day of crossing. Different types of bags are used for the purpose. There are also essential to puncture the paper bags with minute holes to prevent mold the mold formation and also to provide the ventilation for the developing seed. Another one is the crossing. The desired pollen or anther have to be collected from the male parent either in the petri dishes or in paper bags. The collected pollen has to be brushed or dusted on the stigmatic surface of the emasculated flower. After crossing, the flowers are again bagged. And finally, harvest. So crossed seeds F1 have to be harvested after maturity and preserved after drying to safe moisture level to raise the F1 germination. So crossing techniques here. The emasculated flowers can be pollinated in different ways are there. When the flowers produce small quantity of pollen, it is the transferred with the self of camel hairbrush. When the flowers produce large amount of pollen, it will be collected in glazed butter paper bags and dusted on the emasculated flowers. In case of clipping method of emasculation, the individual mature anther may be collected and inserted into the emasculated spiclets. When the flowers are processing numerous anthers, individual flowers is collected after the corolla and the calyx are removed and will directly made in the contact with the stigma of the emasculated flower. So the inflorescence of male and female parent are enclosed in the single bag and allow for the pollination. The seeds from the female parent are harvested and raised in the nursery based on the marker gene character. The F1 seedlings are identified. So this one is there. And modification of sex. So that is the hermaphrodite flowers. That is the both male and female reproductive organ in the single flower. It is a complete flower. Here, total five reproductive organ: sepals, petals, androecium, and gynoecium is present. Incomplete flowers means missing up to any one of all. Imperfect flowers, perfect flowers are there. Monoecious and dioecious flowers are also present. So monoecious means sexual. These flowers are unisexual and present at different positions on the same plant. Example, cucumber. And dioecious means male flowers and female flowers are different plants. They are called as a male and female. Dioecious and monoecious. Monoecious means both are present on the same plants. Okay. So here there is sex modification through the hormones and chemicals. So different, which is used in the different chemicals also used for the sex modification. The sex expression is in the dioecious and monoecious plants is genetically determined and which is modified to considerable extent by the environmental and introduced factors such as mineral nutrition. Okay, photoperiod, temperature, phytohormones. Among these, phytohormones have been found in the most effective uh, agents for the sex modification of their role of the regulations of sex expression. Morphological difference in various sex types, their specific metabolic characteristic result from the possession of a specific pattern of proteins, enzymes, and other molecules. Modification of sex expression is cucurbits have been introduced both the changing the environmental condition by applying the treatment with some hormones. Example, auxins, which is auxin treatment, which is increase the female sex tendency, while gibberellins cause the shift towards the maleness. Hormones and chemicals inducing the femaleness, which are the auxins. Auxins means naphthalestic acid, ethanol, ethaphon, cytokines, that is brassinosteroids. These are to inducing the female flowers and inducing the maleness chemicals and hormones, that is the gibberellins and silver nitrate, ABA, thioponic acid, and paclobutrazole. So these one to inducing the maleness flowers. In cucumber, what happened here? The cucumber, the silver nitrate found to be potent inhibitor of silane action, leading to the femaleness. It should be sprayed when the first true leaf is fully expressed. So in the gibberellic acid spray will lead to the excessive elongation and the weakening of plants, and they will be increased number of male formed male flowers with the less pollen. In gynoecium cucumber, there will be increased the number of male nodes when they spread with the silver nitrate and gibberellic acid with the made of position and multiplication of gynoecium hybrid seed production. So some environmental conditions also. Environmental sex modification here due to the production of photosensitivity varieties, mode of era of agriculture, that to develop the sex expressions. In the cucumber, high temperature and long day length the favor male flowers, or either high temperature will be extend the flowering of male flowers. As the temperature increases from 19 to 23, the node first male flowers has also increased from 9 to 16 in number. This clearly indicates the high temperature favor the male and delay the female flowering. The male flower expressions of several plants, this is flowered by high temperature and female sex expression by low temperature. The male sterility mutant of tomato development male sterility, and the minimum temperature is 30 degrees. The normal flowers at lower temperature in process and spores of low temperature effect on the development of androecium. This is the process. And seed development. Next one is the seed development and heterosis. What is the seed development here? The first division of zygote is transverse in the dicots and result the small apical cell and large basals. So after the cell division vertical to form two cells undergo a transfer result from superior upper two superposed cells and result T-shaped core embryo pro-embryo is called. That is heart-shaped or T-shaped embryo will form. The cells and its derivatives under vertical divisions forming a group of 4 to 6 cells. This group divides by uplift periclinal wall forming a set of inner cells and a row of outer cells. The inner cells form the shells of root apex and the outer cells form the root cap. The two cells formed as a result of division again divide vertically from the quadrant. Each cell of the quadrant divide transversely and thus as octagon containing the two higher of the cells. The cells of an octagon undergo vertical division resulting in pro-embryo. Periclinal divisions occur in the peripheral cells of globular pro-embryo that in the outer layer, the dermatogen. In monocotyledons, what happened here, the cell remain divide and develop into the half story of suspension. The cell divides into the transverse division and gives rise to two parts. The terminal cell of these two by the repeated divisions in different planes gives rise to a single cotyledon. The embryo development into classes different from the out of the monopod. A dorsiventral symmetric established as a result of peculiar oblique position of cell walls early in the embryogeny. The single coordinating is reduced to other two and additional structure like polyoptile and polarizer are found. So endosperm here after fertilization, that is the endosperm is developed. So endosperm here three types of endosperms are there: nuclear endosperm, cellular endosperm, allovial type of endosperms. There are three types of endosperms. Seed coat. So seed coat segments oval undergoes recognition and historical changes during the maturation to from the seed coats. In bi-integument oval, seed coat may be diverse from the both integuments or outer integument only sometimes. Inner integument may be dissipated. Seed growth and maturity. When the soybean is a present in monocotyledon, dicots to be stated chance in the pattern of fecundation reserve material in the different stages of the seed maturation. The water content of grain maximum between 14 to 21 days after anthesis, then it is declined rapidly. The amount of reduced sugars and fructose are highly between 7 to 14 days. The speed of germination is faster wheat varieties that is begin to last water early during the seed development. The seed is said to have the physiological mature only when it is attained the maximum driving. and germinability and vigorous. Normally, the seed is harvested at the field maturity stage when the moisture content is reduced to about 6 to 10 in wheat. Field maturity is a crop specific character. The next one is the hybrid seed production. Discuss hybrid seed production. Yeah. Okay. So in the seed development, what here the soya bean means glycine seed attain the maximum dry weight between 48 to 54 days after flowering. So oil accumulation is less during the 12 to 18 days after fertilization. The maximum oil accumulates between 24 to 48 days after flowering. So, which is the rate because the protein content is seen in maximum during the 12 to 18 days after fertilization and decreases subsequently. The initial high percentage of the protein may be due to high content of non-protein nitrogen, which decrease the seed age. So oil accumulation picks up on only after protein accumulation completes the seed. So, today this one is the different topics we are to discuss. That one, not here folate structure and structure of megaspore and sporogenesis, types of ovules, and hybrid seed production. Okay. So thank you everybody. Thank you so much.